Hello, my name is Anne Martel. I'm a senior scientist at the Sunnybrook Research Institute in Toronto, Canada. And I'd like to thank Dr. Berger for giving me the opportunity to talk to you about AI applied to digital pathology. First, some conflicts of interest. I'm a co-founder, shareholder and CSO of PathCore, a digital pathology software company. I also receive research funding from a variety of government agencies in Canada and the US. Okay, so pathology is something many of you are very familiar with, no doubt, but I'll just go through the workflow for those that aren't. So first of all, we take tissue. These are surgical resection samples from breast cancer surgery, but we may also have biopsy specimens. And then we take this tissue, cut blocks from the tissue, embed them in paraffin, and then take very, very thin slices from this on a microtome. These slices are stretched out onto a glass slide and then stained with some kind of reagent that helps produce contrast between different cellular types and so on. And then traditionally a pathologist would look at these slides under a microscope and then make their diagnosis based on that. And they can use different objectives in order to increase the zoom factor as they look through the microscope. Early forms of digital microscopy involved actually mounting cameras on the top of the microscope so that you could just take a photograph of whatever the pathologist was looking at. But more recently, whole slide scanners have become available. For about the last 10 years, it's been possible to feed in a whole tray of glass slides and then output a bunch of very high resolution, very large digital files. These are becoming much more common, and in some parts of Europe, particularly the Netherlands and the UK, entire pathology departments are now putting all of their work through digital slide scanners. The US and other areas lag behind this a bit, but I'm sure over the next 10 years we'll see an increasing adoption of this technology in the same way that radiology went digital some 10, 20 years ago. So the first applications to find their way into FDA-approved products were those that could improve the reproducibility of scoring or grading. So these are basic counting or quantification tasks. For example, in an IHC image, what proportion of cell nuclei have taken up a particular stain, what fraction of the cell membranes have been stained, and so on. So applications for doing ER or HER2-neu stain quantification have actually been approved for clinical use. More complicated is workflow. So if we want to be able to identify which slides could be set aside for reading later to triage them, or which slides need less attention from the pathologist, we need to be able to detect whether there are any abnormalities in that slide automatically. This is very complicated digital pathology because it's quite hard to determine whether or not there are no for example, a single cancer cell in an image might be quite difficult to detect. And in order to get 100% sensitivity, you tend to get very poor false positive detection rates. Of more recent research interest and growing amount of interest in the community are how to create prognostic models and how to integrate digital pathology images within imaging and genomics. So these are more research-based questions, but these offer some exciting possibilities in the future, which I'll talk about nearer the end of the talk. So the use of machine learning to analyze digital pathology images is nothing particularly new. In 1966, Judith Pruitt actually outlined the three basic components needed for pathology image analysis. So the first is a means of creating a digital image. In her day, it would have been a camera attached to the end of the microscope. Now, of course, we can use whole slide scanners much more efficiently. You also need some human analysts who can tell you what kind of features you need to look for. So for feature-based machine learning, you want the pathologist to advise you on what kind of features to extract from your cells, for example. If you're going to do things with deep learning, you still need to have human architects to decide what kind of network you want. You need pathologists to tell you what the problem is that you're trying to solve, to prepare the material that you want to use to train the network, and so on. And you also need a computer which can take the huge amounts of data from these optical images and then turn these feature vectors, of however we got them, into a classification or segmentation, whatever we want. So Pruitt was using this approach to determine the difference between lymphocytes, monocytes, neutrophils, and so on, using a few straightforward features such as the nuclear area, the cytoplasmic area, and the contrast between those, and then feeding this feature vector into a standard machine learning algorithm. And this workflow that was used back in 1966 is very similar to a workflow that was still in use some 40 or 50 years later. So this is a patch from a project that we had in the lab where we were trying to quantify the percentage cellularity of cancer cells that were left after neoadjuvant chemotherapy in breast cancer. So the task is to determine what fraction of this patch is occupied by cancer cells, which are these larger nuclei here, versus lymphocytes and other stromal material and so on. So the workflow is as follows. First of all, we do something to help normalize the color space and improve the image contrast, so manipulating of the color scales, particularly important in digital pathology because the amount of purple and pink hues varies quite a lot from center to center and staining batch to staining batch, so it's something we need to normalize. Then you can use some standard image processing techniques to threshold the image and then segment the cells, so you end up with segmented nuclei. And then in the same way as you do radiomics, you can do pathomics, so you extract a whole battery of features representing shape, intensity, texture and context from the image, and feed those into the machine learning algorithm of your choice. In this case, we use support vector machines to tell the difference between lymphocytes and then malignant epithelial cells. This worked pretty well for this project, so Mohammed was able to actually classify lymphocytes versus malignant cells or malignant cells versus non-malignant cells quite accurately, and was able to get a decent measure of percentage cellularity on these tissue types. But the method's very slow, so if you want to do this across a whole slide image, you have to segment millions of cells and then extract features from each of those cells and then feed it through. So this is unrealistically time consuming. It would take days and days to analyze a single slide. So just 1 year later, and I finally converted the lab onto deep learning methods. So this is a student who took the same data and simply used a fully convolutional network. So here the network was simple. He just took a 32 by 32 patch and then moved it across the image, I think a few pixels at a time. And then for each position of the patch, he trained a classifier to say, is there a nucleus in this patch or not? So you can get a nice heat map of the probability that any patch contains nuclei or any pixel contains a nucleus. And then you can train the same network then to tell the difference between lymphocytes, normal epithelial cells, and malignant epithelial cells. So it turns out that using the deep learning approach is much, much faster than using a conventional feature-based approach and is actually more accurate as well. So of course, as in radiology, the same has happened in digital pathology since 2013, when Sirisan was actually using deep learning methods to look at electron microscopy and mitosis from Schmidhuber's lab in Switzerland. Since they won the first challenges in these areas using deep learning networks, the whole field has basically swung that way, and there's been an exponential growth in papers on computational histopathology using deep learning. And Chetan, one of my postdocs, has actually written a nice survey paper on this, which should be published in Medical Image Analysis soon. And we can see from the categories of what kinds of methods are being produced, it's very similar to medical imaging. So much interest in classification. These were the first things that people try to do with deep learning. And then we get on to things like segmentation. Transfer learning, particularly important in digital pathology to take care of the staining variations. And then more recently, there's been a lot of interest in weakly supervised, unsupervised learning, and also in survival models. So for this talk, I'm going to concentrate more on why digital pathology is different to medical imaging. So although we have the same kinds of tasks, the actual way we implement these has to be informed by the particular properties of digital pathology images, and I'll go through those next. So the first big challenge we have in digital pathology is one of scale. So this is a digitized whole slide image of a DAPI stain in liver cancer, and it was digitized at 20 times magnification, and this one slide has 35,000 by 45,000 pixels, about 1 gigabyte in size. So clearly you need to have some kind of pyramidal representation of this. You can't expect to load in a gigabyte image onto your desktop in MATLAB and expect your software to work happily with it. So typically you use a pyramidal format to sort these images so that On the first level, you have some kind of thumbnail, so you can quickly get a rough idea of what the slide represents. Then you can go down a level and get a little bit more detail. If you go down to level 2, you can see that we can't fit the whole slide anymore on the screen. We're now at over 2000 by 2000 pixels for the whole slide image, and over 130 tiles now in that image. And if we go down to the next level, down here, we can see that we're into even larger images now, and this is where we come to the full resolution of the image. So to show the whole image on the screen, we'd need to have 32,000 tiles. So this image here is just this small area on this slide here. And the problem gets even worse when we start to digitize at higher magnification. So this is 40 times magnification, which is what we're working with to doing breast prognostic models. If we pull out tiles from this image, so if we want to cover the entire image by tiles, the same size as the 224 by 224 tiles used for ImageNet, we get 220,000 tiles. So just 6 whole slide images gives us the same amount of pixel data as the entire ImageNet database. Obviously 6 images is not sufficient to train anything. We need to represent more variability between patients, between cancer types and so on. So typically we need 100 or even 1,000 or more images. So you can imagine what kind of issues we have when dealing with data sets that are 1,000 times as large as ImageNet. So another challenge is in annotating the image. We can't just use the standard diagnostic radiology tools for annotation. We need to use a proper slide viewer where you can zoom all the way from high resolution down to low resolution and all points in between. Pathologists really don't like it if you give them small isolated patches to label, as they need to see the whole context around the structures that they're looking at. This is the sitem viewer. It's developed by my group some time ago, and it allows you to produce annotations, label the annotations and so on. And it's currently been funded by the NIH ITCR to add plugins to this. So I'm collaborating with some people from the US to try and add some analysis routines to this viewer. In addition to the scale being a problem when you're annotating, there's also a challenge of inter-observer variability. So annotating pathology images is not something you can easily train graduate students to do. You really do need experts, but even the experts often disagree. So these are 2 pathologists here who were asked both to annotate ductal carcinoma in situ so that we could train a segmentation algorithm. And you can see that one pathologist here has labelled these ducts in blue as definitely DCIS, and these ducts in yellow as probably DCIS, whereas this pathologist thinks that these are all definitely DCIS and these may be also DCIS. You can also see there's a difference in level of detail. This pathologist has gone at fairly low resolution and has gone around the outside of the ducts, whereas this pathologist has gone all the way around the inside. So this is the kind of inter-observer variability that we have to wrestle with. Another challenge in digital pathology is focus. So if we have thousands of images in a single whole slide, thousands of patches in a single whole slide image, it's very computationally costly to feed all those through a training algorithm. Also, if we feed all of the background patches and all of the fat patches, we run the danger of actually swamping any useful information content that may be in the stroma or the epithelial cells. So there's a lot of interest in digital pathology in attention-based sampling, where you try and be more selective about what part of the images you look at. And there's a whole bunch of different kinds of algorithms to do that, which I won't be going into much in this talk. Okay, so I'd like to return to the challenge of scale with respect to a particular challenge within my lab. So the task here was to segment ducts from ductal carcinoma in situ in breast cancer. So you can see that at low resolution, you can see the outlines of the ducts very nicely, the stromal tissue around. So we'll be able to get good segmentations of ducts, but we don't have the detail to know whether the duct is normal or malignant. At high resolution, we can actually see whether the cells are malignant or benign, so we can tell if this is DCIS or whether it's a normal epithelial, a normal breast duct. But we don't have much information about the boundary, so we'd have trouble, for example, knowing whether this was invasive or non-invasive cancer. And the medium case actually gives us neither one nor the other, so it We can't tell whether it's malignant or benign terribly well, and we don't get a whole duct in one image. So the first question in most digital pathology tasks is, what scale do we extract the slices at? So this is a project done by one of my students, Nikhil Seth, and he took a multi-scale approach. So the first thing you might want to do is a fairly obvious approach, which is choose one of the scales, train a segmentation U-net and see what kind of accuracy you get. So he tried training segmentation U-nets with high, medium and low resolution patches and looked at the dice coefficient with some expert labels. We can try and be slightly more intelligent about that. So another thing he tried was to concatenate the 3 images at 3 different resolutions to form a 9-channel composite image. So 3 times red, green, blue images gives you 9 channels. So then train another U-net, but this time using 9 channels instead of 3. And then a third way he tried to do it was using this 3-armed network, where he actually feeds in the low, medium and high into 3 arms of a U-net. So they each have their own feature extraction legs here, but then you feed in information from each resolution on the upward sampling leg of the U-net. So this was what he called a 3-arm approach. So Nikhil was actually able to get some quite interesting results from this work. He found that the high resolution patches gave quite nice appearing segmentations, but when you look at the dice scores, they were quite disappointing. So here the dice between 2 different pathologists is 0.73, so this is a pixel level quality metric, and the F1 score is more to do with how many ducts are true positive and false positive detection. So this is more of a duct level metric here. The high res only gave a dice coefficient of 66, whereas going down to low res, which in our case was just 5 times magnification, we got a dice coefficient that was actually a lot better, and also the F1 score. So this seems to suggest that actually the context was much more important in finding the ducts correctly than being able to tell the difference between malignant and benign epithelial cells. So the context gave more information. And another big difference is 37 minutes for the whole slide image for the high resolution, because you can see these small patches you have to go over many times and run many patches through a U-net, whereas at the low resolution you can run far more pixels through the U-net at one time. And this only took 6 seconds for a whole slide image, so a very fast method. Then when he looked, the highest F1 score was obtained using the 3-arm method. So this is where you feed in 3 different arms of a U-net with each of the different scales and then combine that information. And you can see that we get very nice appearing segmentations, quite high accuracies. It takes quite a long time, so we're almost at an hour for a whole slide image, which is fine for 1 or 2 images, but we have thousands in our data set that we need to segment, so this is a bit of a problem for us. But it turns out that just taking an ensemble of the low resolution networks, so the low resolution networks had intermediate sort of accuracy, but if you take 10 of these and then take the ensemble output from those, you actually get very high dice coefficients and just a small hit on the F1 score. So you can see that this is the average probability patch of 10 low resolution U-nets and the segmentation here. Now, as well as being a challenge of scale, this is actually a challenge of annotation. So here the blue and the green are 2 different pathologists outlining the DCIS. So again, you can see one pathologist has gone for slightly more detail than the other pathologist, but neither of them have been particularly careful about outlining every single individual duct. This area here in red and yellow is the 2 pathologists outlining an atypia area. So this is not normal, but it's not DCIS either. So the question is, how do we train the best possible network if our ground truth is so inaccurate anyway? So it would take a huge amount of time for a pathologist to draw in detail around every single isolated duct in the training set. But in fact, that's actually what we need if we want to start looking at stromal features and things around the ducts. So this is a big challenge for annotation as well, is the time it might take to do the annotation and the high degree of variability between pathologists. So another student of my lab has attempted to address this by adding classification patches to a segmentation training task. So So the setup is this. We have some patches that have actually had full segmentations done on them. And as I said before, these are very time consuming and expensive to obtain. And most of the publicly available datasets don't contain highly detailed segmentations. They tend instead to contain patches with a single class label attached to each patch. So for example, you might have some training data where each individual duct has been outlined, but you might have other training data where the label just says whether it's malignant or benign inside the whole patch. So what Ozan's done is he's trained a segmentation network first. So in each batch, he takes the patches that have some kind of segmentation label on them and feeds it through a segmentation network. And then he takes the patches that only have labels on them, feeds them through the same segmentation network, and then a secondary classification network, and that propagates the error from that as well. So he found that by using both segmentation patches and classification patches mixed up in the batches, he was able to actually obtain much more accurate segmentation results using a smaller number of segmentation patches if he had a large number of classification patches available. So he's actually submitted this to medical image analysis, so hopefully that will be coming out soon. So we can go one step further and try and train a network without using any labels at all. So the way this is done is we take an unlabeled data set and we find some kind of pretext task that we can carry out without using any labels. We train the convolutional neural network on this pretext task, and then once it's done, we take the learned weights and transfer them to a new task with a very small number of labeled samples. So this is similar to the idea of one-shot or few-shot learning. The kind of pretext tasks that are commonly used in the machine vision community are things like this. So you take an image, you take a center patch for your input, and the task is actually to predict for each of these sub-patches here, where did they come from? So which of these 8 positions did the patch come from? Now clearly this won't work in digital pathology because there's nothing about the orientation of the image that's predefined. But Chetan in my lab, postdoc, is working on pretext tasks that will work with digital pathology images using various aspects of scale and color and so on. So hopefully we'll be able to train ConvNets with very large amounts of unlabeled data and then transfer them to cases where we only have small numbers of annotations. So a very common scenario in digital pathology is where you have a slide level label, but you don't have a pixel level label. So in this case here we have prostate biopsy specimens. This is work by Campanella et al. And the task is to decide whether this slide contains cancer or not. So this is for a triaging application. The problem is that you know there's cancer in here somewhere, but you don't know where it is. So what you do is you break down the image into patches or instances, and for a positive slide you know that some of these instances have to have a positive label, but for a negative slide you know that none of the instances have a positive label. So what you do is you take your instances, you feed them through the model, and then you rank them in order of probability of tumour. Then you take the top ranked instances and use that to generate your cross entropy loss, where you use as the label the whole slide label, and then you use that to train your image. And they showed that if you have enough data, then this works quite well. So they showed that for data sizes of 10 to the 4 images, you could actually get quite good error measurements, whereas if you had much smaller data sets, then this doesn't really work very well. And that's actually what we found as well. You do actually need a lot of data to make this kind of brute force algorithm work. These people had 7 NVIDIA DGX1 workstations and 12,000 pathology slides for prostate biopsies. So this is not something that's very easy to reproduce for most of us in our lab. So we need to be a little bit more intelligent about how we actually try and go about multiple instances. So this is an example of weakly supervised learning, where we're going to try and use the slide level labels to actually train a segmentation algorithm. So the task here is from the Chameleon dataset. This is a lymph node from a breast cancer patient. Some of the slides contain cancer and some of the slides don't contain cancer. So the question is, just knowing that, can we actually train a segmentation algorithm to find the cancer and localize it? Now the problem we have is that we can't feed all of the patches in one of these slides through a single batch. So the assumption that was made in the previous example, where we know that some of the instances in a positive batch have to be positive, is now violated because we may end up with a batch with no positive cases at all. So to overcome this noise problem, Shazier developed a method that incorporated two different loss functions. One was the same as previously, where you look at the loss based on the slide level label, but the other one was to see whether you could incorporate a latent representation of the data. So she used a variational autoencoder to embed the features from each patch into a lower dimensional space, and then she used a clustering algorithm to generate an augmented cost function. And this use of autoencoders or other latent representations to deal with these very large problems with many, many patches in high dimensions is going to become increasingly important in digital pathology. And you can see here that this case here, this is the ground truth label, and this is the segmentation that she was able to achieve without using that ground truth label, just using the slide level labels. So to end, I want to very briefly show you one example of training survival models with digital pathology data. This is work by Lee Cooper's group, where they're trying to predict survival times based on the appearance of the digital pathology images. Here, it's not completely end-to-end. They have to sample some high-power fields that are known to be associated with the tumour. I haven't really got time to go into the algorithm, but it's available in press here. And the reason these survival models are attracting so much interest is because once you've trained a survival model to predict outcome based on the whole slide image, you can then start to decide what part of the image drove that decision. So here, this is a survival model that's been trained on whole slide labels, but then they've applied that model to individual patches within the slide. So you now get a heat map where the temperature of the heat map reflects the probability of death. So an area that's bright red is a very aggressive part of the tumour, or that's the assumption. And the reason why that's exciting is because then the pathologist can go and look at the areas that are red and see what's different about them to areas that have a low probability of death at a certain time. So here, for example, they could see that in this particular cancer, infiltration was actually a marker of an aggressive tumour, whereas in a different kind of brain tumour here, edema turned out to be a marker of aggressive tumours. And this is an area where we can start to use digital pathology to try and explore what's driving tumour aggressiveness and so on. So it may actually result in new discoveries. Okay, so I hope I've given you a taster of some of the things that make digital pathology fairly unique compared to medical imaging, and also given you a sense of some of the exciting things that deep learning can achieve when applied to digital pathology images. I'd like to thank all of my group members here, particularly Sharon Nerfik-Moses, who's the pathologist, the breast pathologist I work very closely with, and also invite you to ask me any questions. And this is a plug for MICAI 2020. This is the main conference which talks about medical image analysis. This year we've got 541 papers accepted, the majority of which feature deep learning in some form or other. And because it's virtual, registration costs are actually very affordable this year. So I'd encourage you to go to the website and register for the meeting.